Welcome to the channel. Today we shall discuss how different types of proteins they bind to DNA. Although DNA's primary structure is linear, however, it assumes many different configurations when it binds to its respective regulatory proteins that can be either transcription factors or repressors or activators. For binding these different types of proteins, the DNA possesses different structural motifs in its structure, and it is with the help of these motifs that these regulatory proteins that either increase the expression of a certain gene or decrease its expression, they bind to DNA and then they perform their respective function. So, in order to interact, uh, in order to facilitate the interaction between the DNA and the protein, there must be atomic contacts between protein residues and bases, and protein residues are the sugar phosphate backbone of DNA. So, it's important to remember that many of these contacts, they are in the major groove of DNA, which is also understandable by common sense because the major groove provides a much greater place to sit in for the regulatory proteins compared to any other place in the secondary or tertiary structure of DNA. When we try to characterize the DNA binding motifs of proteins, 80% of them can be assigned to either one of the three classes. These motifs they can belong either to helix turn helix class, they can be zinc finger domains, or they can have leucine zippers. What are these? We should discuss them in detail ahead. But in addition to these DNA binding domains, the proteins also possess other domains that interact with other proteins. This is in case where the regulatory process of gene expression involves a hierarchical binding of a number of proteins one after the other. So in this case, the first protein which binds to DNA will bind another protein, that protein will bind to another protein, so on and so forth. So talking about the helix turn helix or the alpha helices, what they are in the protein and how they bind to DNA. This provides, it's very easy for a linear structure to align with another linear structure. That is, DNA can align more comfortably with a helix in the protein structure rather than with some globular or another structure of complicated type. So a recurring feature of the DNA binding protein is the presence of alpha helical segments that fit directly into the major groove of B form of DNA. B form of DNA is the one commonly found in normal physiological conditions, and we have discussed about it in previous lectures. The diameter of DNA double helix is around 1.2 nanometer, and major groove of DNA is about 1 nanometer wide, 1.2 nanometer wide, and zero point 0.6 to zero point 0.8 nanometer deep. Therefore, proteins they are able to recognize specific sites in the DNA for interaction. The first helix turn helix motifs were the first that were identified, and as we know that many of these discoveries were made first in the prokaryotes. So these first three prokaryotic proteins in which helix turn helix motifs were identified included two repressive proteins and one catabolite activator protein of E. coli. All these three types of proteins they bind as dimers to direct axis of symmetry on DNA. As we have previously said somewhere, that DNA double helix is built on an imaginary axis of symmetry, which we call as direct axis of symmetry. These proteins, each of them contain two alpha helices that are separated by a loop with a beta turn. The C-terminal helix fits in the major groove of DNA, whereas the N-terminal helix stabilizes by the hydrophobic interactions with C-terminal helix. This is the direct axis of symmetry, which is the imaginary axis around which the double helix is wound. This is the major groove of DNA. This is the minor groove. As anticipated, the major groove provides a much larger space for the proteins to settle and interact. And the proteins, they have these helical regions which can directly align with DNA and bind to it. The same class of DNA binding motifs in the proteins is called zinc finger domains or zinc finger motifs. First of them was discovered in transcription factor 3A from Xenopus laevis, and now they are known to exist in nearly all organisms. There are two main classes of zinc finger motifs. One are called C2H2 and one are called CX. The C2H2 domains actually consist of two cysteine residues separated by any other uh, two amino acids and two histidine residues separated by any three other amino acids, and both of these repeats they are separated by at least 7 to 8 amino acids. The CX domains they consist of either 4, 5, or 6 16 residues separated by a pairing number of other residues. This is what a zinc finger motif look like. There is a zinc in the central core, which interacts with cysteines and histidines, and this then forms a structure that can interact with DNA. It can either take the form of a globule or a linear structure. The secondary structure of zinc finger motifs can take the form of a beta strand and an alpha helix that then fits into the major part of DNA. The CX-type zinc fingers consist of two main domains of four cysteine ligands to zinc, followed by an alpha helix. The first helix is DNA recognition helix, while the second helix packs against the first. This is again a three-dimensional model showing how the zinc fingers they can form where a zinc is present at the center and other domains they protrude like fingers. The third category of motifs in DNA binding regulator proteins are leucine zippers. They were first identified in a DNA binding protein in rat liver nuclei, but again, it is also now found in nearly all organisms. The characteristic feature of this type of DNA binding domain is a 28 residue sequence with leucine at every seventh position, which forms a basic region. This suggests that amphipathic alpha helix and a coil dimer will form. This is the structure where there is a leucine at every seventh turn, and this then takes the form of a leucine zipper when taken in three-dimensional form that we will see ahead. Leucine zipper proteins are also called B-zip proteins. They dimerize while they perform their function, and this dimer may be a homodimer or it can be a heterodimer. This recognizes the basic region in the DNA, and this basic region is often modeled as a pair of helices that can wrap around the major groove. When homodimers and when heterodimers form, the homodimers they form and they recognize when DNA has a symmetry. It is a symmetrical DNA has a diad axis, whereas heterodimers recognize even the non-symmetric, non-diad axis DNA. The examples of leucine zippers are FOS and GEM. This is a pictorial depiction of how a leucine zipper can look like. It forms a zip-like structure which holds together the basic region of DNA and links it to the protein. This is a three-dimensional structure showing how a zipper motif can interact with the major groove of DNA. With this, we end a brief overview of these regulatory proteins that bind to DNA and help DNA perform its function. Thank you for your attention.